This meeting is now being recorded.
Okay, I want to welcome everybody to our management discussion series. This is Robert Merrill, Institutional Sales here at Ascendiant. I'll be the moderator for today's call. We'll have a question and answer session near the end of the call. If anybody would like to ask a question, we'll open your lines at that time. Today, we are pleased to be joined by Martin McGlynn, Gregory Schiffman, and Stephen Hun from StemCells. I want to welcome you all and thank you for joining our series. And I'll turn the call now over to our healthcare analyst, Kay Nakai.
Yeah, thank you, Robert. My name is Kay Nakai. I'm the Senior Healthcare Analyst here at Ascendiant Capital. We are happy to have with us today the senior management team at StemCells. StemCells, the ticker symbol STEM, as their name suggests, is engaged in the research, development, and commercialization of stem cell therapeutics and related technologies, and specifically focused on the development of human central nervous system stem cells. We do cover the stock and currently have a buy rating. And, what we like about the company is its demonstrated commitment to rigorous and robust research and development. Given the many unknowns associated with this new field of medicine, we believe that StemCells has been very methodical, maybe more so than others, in its approach to conducting human clinical trials. And while this has resulted in perhaps longer than anticipated enrollment in their respective studies, ultimately we believe that their conservative approach will serve them well in generating the evidence necessary to de-risk their technology. So we believe that now really is a good time for investors to either revisit or become more familiar with stem cells, as we expect to see much more human clinical data from its current and planned clinical studies going forward. The format for this call today will essentially be a fireside chat. And so with that, let's get started. So first of all, I want to welcome the three of you for joining us today. And to start out, for Marty, I guess, can you describe the different types of stem cells, either embryonic, pluripotent, and adult, that are being investigated by your company and others for therapeutic applications? I know we have this broader umbrella of regenerative medicine and within that cell therapy. So talk about your specific product and how it differentiates itself from some of the other alternatives and methods that other companies are developing.
Sure. Okay. Thank you very much, and welcome everybody. So, everybody, I'm sure, is aware of embryonic stem cells. That's where all of the controversy and the discussion, politics, and social and religious tensions that have emanated as it relates to this technology platform. So embryonic stem cells essentially give rise to every cell in the body, and that's the great news. But you cannot directly transplant an embryonic stem cell into a patient or into an organ system in a patient, because they do give rise to every organ and tissue in the body, and in an unregulated fashion they will derive teratomas in the host. So you have to do some manipulation of an embryonic stem cell to extract and terminally differentiate the cell type of interest that you want to transplant into a patient. The next category of cells would be those that are derived from adult tissue, and then those cells are developed, purified, expanded in some cases, and in others not, for transplantation purposes. That is the broadest category of cells, in that you can extract cells, essentially any tissue in a host. The cells can be extracted. Of course, not all cells are stem cells. And many of the cells that are furthest along, either currently marketed mostly in an autologous fashion, or in late-stage clinical development, are cells that have been derived from adult tissue, whether that is bone marrow, blood, fat, etc., just to name a few. These are typically known as mesenchymal cells. And they, for the most part, act like drugs. They don't engraft into the host. Their effect is trophic, and then they're secreted or absorbed into the body. And so essentially they act like a drug. And for that reason, because they don't integrate into the host, the regulatory concerns and the safety issues that go hand in glove with embryonic stem cells just do not go hand in glove with the mesenchymal cell population. They're essentially like a drug. And so the FDA approach and concerns about a cellular agent that does not engraft into the host, they have a different safety profile and a much easier pathway, if you will, through the regulatory process. So for that reason, they are furthest along in the development process, and also because they look and smell and act like a drug, the pharmaceutical industry can better understand them. And so for that reason, some of these technologies have been partnered by pharma. The next group of cells would be those cells that are extracted from a particular organ or tissue that essentially are stem cells. That is to say, they engraft into a host, they migrate and differentiate in a site-specific fashion into the cells that make up the organ system from which they're extracted, and they endure for the life of the host, and they make more of themselves. So those are adult stem cells. And Stem Cells Inc. has a very specific view of definitions of stem cells, and unless the cells meet those criteria, we wouldn't recognize them as a stem cell. Now, the approach that we're engaged in is we extract stem cells, in this case neural stem cells, from fetal brain tissue, and we expand them and grow them up and put them into cell banks, and we keep them in a banked fashion in a cryopreserved form. The approach is allogeneic, that is to say not patient-specific. These cells can be transplanted and used in patients where the procedure is indicated, so whether it's into the spinal cord, the brain, or the eye. The cells do engraft, we know this from our data, do migrate, do endure, and do replicate themselves. So they certainly have all of the hallmarks of a stem cell. They are not embryonic, they are not viewed, or embryonic derived, and they are not viewed as being teratoma forming. The agency views our cells as being non-tumorigenic. So this approach essentially allows us to start off with an active biological ingredient, which are these highly purified population of human neural stem cells that are preserved in stem cell banks and awaiting their application into patients where indicated.
So, if I were to summarize that, it sounds like you're in almost a sweet spot in that you have all of the potential benefits of a stem cell, while also not being restricted as an autologous product. That being said, the ability to engraft, migrate, and survive, I think, has probably caused some conservativeness on the part of the FDA once you went into human studies. So talk about maybe those first two phase 1 studies you did in NCL and PMD, the types of really no-option patients that you did enroll, and actually what you ended up finding in those phase 1 studies.
I will do that, Kay. Let me add to your comment. I think you're absolutely right. I think you've summarized it perfectly well. You alluded to a long process of enrolling patients, and Stephen will explain more about that in a moment. So while the FDA looked at our cells as not being tumorigenic, nonetheless, the mere fact that our cells integrate into the host, they become part of the host and are regulated by the host and endure and survive, that creates a whole different bar and differentiates the regulatory requirements and safety requirements over and above that they would require from cells that are derived from other tissue sources. So I will hand over to Dr. Stephen Hune, who will explain the regulatory paradigm that we encountered and overcame as we prepared and executed our first and second clinical trials, and then how we have gone on to beyond those very rare conditions.
So thanks, Martin. If you could imagine the confluence of elements required to move into the first-in-human study involving a stem cell approach to a central nervous system disorder that would be based on having the cell engraft long-term in the host, and a provocative, innovative technology, first-in-human application, and then the innovation of a stem cell therapeutic lend a lot of gravity toward all of our discussions with both ethics and regulatory bodies. The best way to compare what we're trying to explore is, at a crude level, the success of organ transplantation, solid organ transplantation, the heart, lung, kidney, liver, that we have seen in the last 5 decades now evolve into partial face transplants. But we're not able to do that on a whole organ basis for the central nervous system or the eye. But you can do that at the cellular level. And now that we have a neural stem cell, we have potentially an entity that can be transplanted, again, not the whole organ, but critical parts of the organ, perhaps the most important cell within the central nervous system. So that's sort of the therapeutic premise. And as a neurosurgeon, I draw a lot of comfort from the success that my colleagues have had in solid organ transplantation and bone marrow transplantation. And the last frontier, if you will, in transplantation medicine is the central nervous system. So we've got now a cell that we can begin to explore that very concept. Our first studies were in fatal diseases, ultra-rare and rare diseases both, that have no effective treatment, no effective therapy, and they occur in children. And based on our preclinical data, we believe there was a strong rationale to both explore a fatal lysosomal storage disease known as Batten's disease, or more technically neuronal ceroid lipofuscinosis, but Batten's for short. And that was our first in-human study. It was an open-label study in 6 patients. And because of all the elements that I just discussed, you can well imagine that there was a degree of regulatory conservatism in how we approached to execute that trial. And, the things we had to do involved staggering the transplants. So we had to have at least a month between each transplant. We had to conduct a DMC meeting before each transplant before we would proceed with the following dosing. So that stretches out the time in which you can execute a study, but at the same time you're metering out safety observations before you dose the next patient. There was a dose escalation part of it as we went from 500 million cells to 1 billion cells, which is the most number of allogeneic cells that have been placed into the human brain. We were restricted to a single clinical site to keep our clinical experience and safety observations consistent and uniform. And then there's a whole host of very intense follow-up examinations, both clinically and radiographically, that were required by the regulatory body, as well in terms of good clinical design test this first therapy. So we've now emerged from that paradigm of staged transplants, DMC oversight at a very, very intensive level, heavy regulatory reporting and updating, single study trials now into diseases that have much larger unmet need, where we're now able to conduct multi-center studies without the same restriction in terms of dosing frequency in our patients. So we've gone from intensive safety scrutiny as we test something that's really a first-in-human application to now moving in a much more expeditious fashion as our clinical experience has evolved. We were able to glean many important clinical and scientific objectives in those first two open-label studies. Despite the fact that they were in rare diseases, it's a little bittersweet because in a fatal disease that's progressive, you will have the opportunities to look at post-mortem tissue. And we've done so in the NCL trial, in which patients succumb to their disease as expected. And we had the opportunity in the NCL study to determine whether the cells had survived. So the very attributes that Martin had described of engraftment and migration, we think are a foundation of how the cells will be efficacious in the brain and the spinal cord and eye, that they'll survive, they'll have a long-term impact after a single application. And we were able to show in the NCL trial that the cells, in fact, did survive, in one case up to 2.5 years after transplant and 1.5 years after stopping transplant, after stopping immunosuppression. So that was a very important observation for us, to see that the cells had survived. And not only that, but we could demonstrate the cells had migrated from their original site of deposition in the brain. So now we're beginning to see evidence in the human that replicates the animal data. And that's very encouraging at this early stage in translation. The next study that we did, the study in Pelizaeus-Merzbacher disease, which is a rare disease involving myelination. And myelination disorders are diseases that affect the insulation or the sheathing around nerves or the axons. And this is a disease that children suffer lack of normal myelination development, so they have tremendous white matter abnormalities and the consequential failure to develop normally, both from a motor and a developmental standpoint. And they can be diagnosed very early on. And the advantage of the second trial that we did was we had an MRI way of looking at cell engraftment, because if we detected signs of new myelination as a result of the cell transplantation, which is what we expected based on our preclinical studies, that we could argue that there was evidence of efficacy and a biological effect in the second trial, which again was for PMD. And so what we gleaned from the second trial was in that very fact that we could see in all the patients there was evidence of changes in the MRI scan that corresponded to where we had transplanted the cells, and the changes corresponded to increases in myelination in those regions, which we've interpreted to be signs that the cells have engrafted and are producing myelin in a myelin-deficient disease. So, we had a steep learning curve in terms of trial execution, regulatory interaction, but we've gotten through those first two trials now with a lot of very important information, which has really encouraged us to continue moving forward into much larger diseases and more complex unmet need.
Okay, so I think without minimizing in any way the amount of work that went into those studies, I think some key takeaways are clearly, one, safety, the ability to inject up to 1 billion cells into patients and not see any adverse events into the cells themselves. And then number two, a good concordance in terms of the translational research from what you saw in animals and then what you actually saw in humans.
Yeah, I think that's a very good, absolutely.
Okay, so with that in hand, you've now progressed into phase 1/2 studies in both thoracic spine injury and AMD. So let's talk a little bit about the thoracic spine injury clinical trial. Obviously, you had some good news just the other day about completing enrollment in the study. But can you talk about the study, just kind of give the general parameters, and then also talk about some of the interim data that you've presented thus far on certainly the first cohort and what we could expect to see as far as a next data update from the study?
Sure. So spinal cord injury has always captured the imagination of scientists for a cellular transplant approach. And there is a lot of preclinical data that suggests that this is a very reasonable approach to pursue. And we have very good preclinical data in an animal model of injury that when you place the cells at certain time points after the injury has occurred in the animal, that you see evidence of the cell migrating and differentiating into both neuronal phenotypes, which is the main cell, as well as oligodendrocytes, which are the cells that produce the myelin, which could, in essence, encompass a number of mechanisms for why spinal cord injury patients suffer the deficits that they do, because it's absence and crush and destruction of neurons, as well as oligodendrocytes, or the myelin within the spinal cord. So it certainly captured our imagination to move as we developed our safety, feasibility, and tolerability from our first 2 open-label studies to now begin to ask this question in a non-fatal disorder, if you will. And it was important for us to execute a trial that was a departure from the normal paradigm of how spinal cord injury studies are done, at least the first-in-human studies. And by that I mean, classically what's done is you start in thoracic injury because it's less risk than cervical injury in terms of an intervention, particularly surgical. And classically, you're restricted to patients who only have complete injury, so no motor or sensory function below their level of injury. So we understood that, but we also felt it was important to include in our first trial patients who had incomplete injury. So those are patients who have some degree of continuity of function across the level of injury. And if you can imagine, then those are patients who probably have a better regenerative potential because they have not had a complete insult to the cord. So our first study in thoracic spinal cord injury was designed to include both complete and incomplete studies. And we, again, moved through the complete and incomplete on a risk-based assessment, first getting experience with those with complete injury before we moved into patients with incomplete injury. And as you said, we've now completed that trial. It involved 12 different subjects. And the other element of the study that was important for us, and one of the reasons why it was done in Switzerland, is because we wanted to get a very close assessment of what the level of function was, both above and below the injury, with very sophisticated electrophysiological and clinical assessments. And Dr. Armin Kurt, the PI in Zurich, leads a center where they're particularly adept at doing these assessments. And that's important because we wanted to do 2 things. One, to make sure that we weren't disrupting the segments, if you will, the little individual segments of the spinal cord above and below the injury where we were transplanting cells, but also detect signs of efficacy, whether we were seeing changes, which can sometimes be subtle, but again, that would correspond to a biological effect. And so we're now beginning to look at the data from the study, and as you alluded to, in the first 3 patients that we enrolled in the study, all of whom had complete injuries, we saw some unexpected findings. And those unexpected findings were that patients who were now many months post-injury, and 2 of those 3 were able to have evidence of sensory gains well below their level of injury. And sensory gains that involved more than one segment of the spinal cord, sensory gains that involved more than one modality of sensation, and that they were persistent. So this was very intriguing to us, and that it corresponded also to the electrophysiology recordings that we're able to do across the spinal cord injury as well, which is a highly objective measure of cord function. So for us, this was very intriguing to see, certainly from a safety standpoint, very, very reassuring, but also the signs that maybe there's a biological effect of the cells. So this was extremely encouraging for us to see in the study, And we're going to be looking at the data from the remaining patients in the study, and we expect to have an update with further interim analysis in mid-May of this year with the PI from Dr. Kurt at the American Spinal Injury Association meeting in May.
Okay, great. So obviously the encouraging results that you saw lead to more questions, and I think ones that I often get are, one, how do you measure and qualify in terms of a benefit, improvements in sensory versus improvements in motor activity? In other words, the dream obviously is for that patient who's in a wheelchair to be able to get up and walk, but falling short of that, what types of actual achievements and improvement in sensory or motor function really would be viewed as significant achievements?
So it's a great question, and it turns out that if you interview and talk and poll patients with spinal cord injury, and you ask them what would be a meaningful change, you don't get the response that we as people with intact spinal cords would think. That it's not so important for them to be able to get up and walk. But there are other things that are incremental that do have an impact on their quality of life. And I'm just saying this as a way of putting it into context. That we understand spinal cord injury is a very challenging disorder to try to pursue. But, a cellular approach could well offer incremental gains to a patient with an injury that could have meaning for their quality of life. And I'll take you through some examples. The simple return of bowel and bladder function could be a benefit to a patient. The ability to stabilize certain autonomic functions, heart rate, blood pressure, which is a consequence of spinal cord injury, would have benefit. Return of sexual function could have benefit. And then, in addition, what I think is eminently achievable based on our understanding of the technology is, can we gain a patient an additional muscle segment or motor segment in their arms or legs? So if they have weak elbows, can we give them some wrist strength? Can we take their intact wrist strength and give them better hand strength? If you can do that, which although an incremental biological effect could take someone whose inability to drive, say, and make them independent because now they have better use of their arms, that's a change in quality of life for a patient with spinal cord injury. So I think there might be a misperception that we're all walking around thinking that simply putting in stem cells is going to make a patient who can't walk with a very severe injury to their cord. I think that's not our expectation, although it certainly is a goal. But I think there's a lot achievable that a cellular transplant can do for these patients that would have clear clinical benefit for them and change their quality of life. And based on our animal models, that's why we believe it's important to now move into the cervical study, where you can ask a much more complex question because you can get to whether we are assessing changes in motor function. So the very thing of changing elbow strength and wrist strength and hand strength is something that we're going to be able to measure in a very ready fashion with standard validated hard clinical endpoints in our upcoming study that represents the evolution of the spinal cord program here at StemCells.
So I think that leads into where you're going next with the program, which is to conduct a phase 2 proof of concept study in cervical spine injured patients. And I think the first question is, do you have an ideal patient who you theorize would be most responsive to this type of therapy? Obviously, a cervical spine injury not encumbered with the amount of damage that perhaps is associated with a thoracic spine injury, but also higher up on the spinal cord and possibly affecting more of the lower body. So can you give us some thoughts on what an ideal patient candidate might be, also not only in the area of injury, degree of injury, but also how recent the injury was.
So, we're still learning about what we think might make an ideal candidate, but if you look at our animal data, it would suggest that it's someone who's between 3 to 4 months after injury and between 1 and 2 years thereafter. So there's a window of time that I think there's regenerative potential for the spinal cord. We don't think that window is immediately after the injury. We think the patients need to recover medically and emotionally. They need to reach a stable neurological plateau for us to be able to measure whether we've got a change in the natural history of their recovery. They have to have, an injury that in many ways may be more incomplete. Now, if you can think about it, a thoracic spinal cord injury means that you've had to have deformed the body to create an impact to the cord at the thoracic level. Cervical injuries tend to be milder injuries because it doesn't take the same level of injury or energy to damage the cervical spinal cord. So we're dealing now with, I think, a less severe injury. At the same time, patients who have motor preservation that we can measure above the level of injury and below the level of injury. So the patients that we're looking for, the ideal patient is someone who's got some degree of continuity of function across their level of injury. So they've got some remaining tissue in which to regenerate with the stem cell approach, who's somewhere between 3 months and 2 years after injury, that window of time, and someone whom we can then assess whether there's been a neurological change, a hard change in their motor function of their upper extremities, that we can say, listen, they're gaining motor function in a real way. That's the result of the stem cell transplantation, which is the point of conducting now, moving from an open label study in thoracic injury to a randomized controlled study in cervical injury.
And so just leading into that design of the next study, I know it's not finalized yet, but what other design parameters can you share with us? And second to that, having described what you think an ideal patient is, how easy or quickly do you think you can identify that type of patient and therefore enroll the numbers you're thinking about in this next study?
Yeah, so to some of this I can speak to, but the cervical study will involve dose escalation, which we think is an important question to ask in a study that's designed to look at efficacy. So we'll have an element of that study that will be dose escalation. The advantage of a cervical study, in addition to some of the things I've mentioned, is that the majority of patients with spinal cord injury have had their injury at the level of the neck, of their cervical spinal cord. So out of every 100 spinal cord injury patients, about, maybe 70% of them have had injury to their cervical cord versus thoracic. So the rate of enrollment we expect to be much quicker than our thoracic study because the prevalence of the disease is much higher. The other difference here is now, again, as because of regulatory parameters, we're now moving from highly intensive safety-based studies at single centers into multi-center studies. And so the phase 2 study will be North American. It will be multi-center. So the combination of a multi-center study with a patient population that's much more prevalent than thoracic is really going to help support accrual and enrollment in the study.
Okay, great. So just before we move on to AMD, to wrap up spinal cord injury, the next data update will be next month, and you plan to initiate the phase 2 study. What's your timeline now for that?
We plan to initiate the phase 2 study this year, in the second half of the year.
Okay, great. So let's move on to AMD. Here we're looking at an eye disease involving the retina. And again, as you started this study, since we're, having moved on from the initial rare fatal disease population to one that, certainly isn't in that camp, you had some very tight enrollment criteria. So can you talk about, again, the basic design of that study and where you're at now in terms of enrollment? I know you're getting close to completing that.
So again, the steps for the AMD program and trial really reflect the same experience to a degree that we had with dosing the brain and the spinal cord, in that because this is, if you will, the first in-human application of neural stem cells for the eye, we had a study that was split into two different cohorts, with the first cohort, because of risk mitigation, being patients who have very severe vision loss. This is a normal regulatory paradigm as you begin to explore an innovative approach that involves the invasive application. So the first 8 patients in the study have vision that's 20/400 or worse, so they're legally blind and beyond. As we tested the cells at a dose escalation in the first cohort, so that's 8 patients who have vision 20/400 or worse, and then we started at a dose of 200,000 cells in the first 4 patients, and with safety and tolerability, which we demonstrated, we then moved into a higher dose of 1 million cells. So now we've gotten through that cohort of 8 patients that have essentially very severe vision loss as a result of their dry AMD. And with the safety and tolerability that we've seen in those 8 patients at both doses, we'll be moving into the second cohort of the study, which are patients who have better vision. So again, this is akin, if you will, to going from patients with complete spinal cord injury to incomplete. So perhaps a patient, because they're at an earlier stage of their disease or less severe disease, has more regenerative potential. So they have, in essence, more photoreceptors left to rescue by our particular approach. And I think, again, it's easy to get confused in cellular transplant modality as what we're trying to achieve for any of our indications. For AMD, what we had recognized was in our preclinical studies, we saw evidence that the human neural stem cell will protect, if you will, host neurons. And it probably does through a number of trophic mechanisms and other physiological aspects of a stem cell. And we were intrigued enough by that to wonder, well, could we have the same effect on photoreceptors, which, if you understand, are nothing more than sensory neurons. They're part of the central nervous system. And so we linked up with a very good scientist at OHSU, Dr. Ray Lund, and asked that question in an animal model of retinal degeneration. And what we found was that when the cells are transplanted, they survive, they migrate, they do the things that we see them do in the spinal cord and brain, but they also ended up protecting the photoreceptors from degeneration, and they preserved the vision of the animal out to 7 months. So we have both a behavioral component and a functional component that mimics the rescue of the photoreceptors. So dry AMD is essentially the final stage of that disease, is loss of photoreceptors. So might we be able to preserve photoreceptors through a one-time transplant for a very durable effect? 5 years, perhaps beyond, would have a lot of meaning for a patient with dry AMD to be able to hold them at their level of visual acuity, particularly if you intervene at earlier stages of the disease. So that's the essence and the rationale for the approach for dry AMD, which, as we all know, has no current effective treatment or therapy, and for which makes up the majority of patients with AMD. A fraction of AMD patients have wet AMD, which is treated through the anti-VEGF treatments, but those patients all mostly start having dry AMD first. So we're very intrigued by an approach that could have a sustainable benefit for a patient with dry AMD and maintain their vision at that level. And that's been the whole rationale for the phase 1/2 study in AMD. And now we're very pleased to be moving into the second cohort of the study. And because, again, with the paradigm of moving from a single center study into multi-center now with 5 sites available for our current study, we expect to complete enrollment of the second cohort of that study in the second quarter of this year.
Okay, great. Yeah, it's nice to see that moving along more quickly now. So in terms of data from the first cohort, when can we see? When can we expect to see the next reporting of that? And maybe just as a caution, given that they are the condition of their site is so bad to start with, should we really just be looking to see safety and not much of efficacy for those patients?
So we've not yet had any public disclosure of the interim data from the ongoing study, apart from the fact that we have ongoing safety. And as I said, we expect to complete enrollment in that study the second quarter this year, and we expect to have an interim update about the status of that study in the middle of this year. The final data will come out, a year following the last patient out, which will be, again, about the middle of next year. I think it's a fair point to say, what can you say in an open-label trial when you've had to start with, because of the regulatory paradigm, patients who have very severe vision loss, perhaps vision loss beyond the regenerative potential. And certainly the safety of the approach and feasibility and tolerability with follow-up of the first 8 patients that we're assessing is a very important first step in the field, particularly something as innovative as our administration and our approach. So that's a very important milestone for us. But I also think that the data that we'll be able to glean from these first 8 patients, at least the interim data, will be very informative going into our second trial, in part because the eye has a real spectrum of objective measures that are easily done, easily repeated, and it's a different type of platform in which to test our technology than that posed by the brain or spinal cord. So we expect, as I said, to release the interim data in the middle of this year on the first 8 patients in the study.
Okay, and I know that the data will drive this decision, but in thinking of the next phase 2 study, at least in theory, what does that ideal patient candidate look like?
Well, that's the importance of our safety and tolerability of this first study, is that we'll have confidence moving into patients with better vision at the time of entry into the study, based upon our approach of stabilizing vision. So I think that's one important aspect. The ideal patient is someone who's probably got vision perhaps even better than what we currently have in our ongoing study, but we're still looking at what that ideal patient will be and thinking about the design of that particular study. That study will be a multi-center study. It will be a larger number of patients that will be designed to look at efficacy. But, the AMD is such a huge unmet need. It's such a prevalent disease that's only getting worse with time, that we're very hopeful that that study will accrue very quickly as well.
Okay, if I may just add to Stephen's comment, for the phase 2 study in dry AMD, we would hope to target the patient population that we would actually hope to that would benefit from the intervention, and that would be subjects for the therapy in the commercial phase. So unlike the earlier stages where we were targeting those patients with the most severe visual acuity, the phase 2 study would be targeting to really recruit patients into the study that in turn would be the population of patients that we would intend to treat.
Yeah, very good. So I think it should be clear to folks that in both spine and AMD, we've gone from the most severe to now a targeted population. We're seeing increases in the rate of enrollment and actually completion of enrollment of the spine study. And, at the end of the day, it's a lot more patient data that people can look at and place their bets on your particular therapy. So I think that's all very positive. Switching gears here to one of your earlier stage programs, that being Alzheimer's. Let's talk about that a little bit. Obviously, you're one of the rare public company recipients of a California Institute of Regenerative Medicine grant, nearly $20 million, to conduct preclinical work in this area. So can you talk about your program there and maybe specifically, what are some of the deliverables that you're looking to achieve over the next couple of years in order to receive the entirety of the nearly $20 million grant?
So, Kay, the $20 million, it's just under $20 million, is not in the form of a grant. It's in the form of a forgivable loan to the company. Now, what that means is if in the course of our March to filing an IND, we hit a no-go problem, if we hit a technical issue or some reason essentially to question the rationale of going any further, and the program is terminated by agreement, then any and all monies received by the company up to that point would be forgiven by CIRM. That said, we are on track to successfully deliver what we signed up for as the quid pro quo for receiving this funding, namely the filing of an IND, which will be the first ever cellular transplant intervention in Alzheimer's patients. The goal of which would not be to per se cure Alzheimer's disease. The goal of which would be to enhance memory and performance of memory function and task. I want to make that clear. The deal we entered into with CIRM was that we would file the IND within 4 years. We are targeting to file that IND actually a year earlier, and so that would mean we would be looking to file an IND in Alzheimer's in the first half of 2016. The objective of this exercise is to essentially be able to replicate what Dr. Frank Laferla and his colleagues have shown in animal models. And Dr. Laferla's lab is at UC Irvine. They have shown, and they published data demonstrating that animal mouse human neural stem cells transplanted into different animal models has a very definite beneficial effect on memory performance. We collaborated with them to see if we could repeat the same outcome using the human neural stem cells into these same animals, and the answer was yes. We were able to replicate his results using the human neural stem cells. And then we formed a collaboration which culminated in our filing an application for CIRM to fund the IND-enabling activities. So far so good. Everything is on track, and in fact, if we continue along the pathway that we're on now, we could well file an IND a year earlier.
And Marty, is it a single preclinical study that's really the key thing you need to conduct, or is it a series of preclinical studies?
Well, actually, what we're doing is we're replicating the work that was done in Laferla's lab, and now we're doing it to confirm the results and to validate what was seen and published in the papers, and to do so in a more controlled fashion as laid out by the research program agreed to with CIRM. And the work is focused on two different animal models. One is known as the triple transgenic mouse, which is, there really isn't an animal model for Alzheimer's disease, but there are animal models where you can generate the plaque, and there are animal models where you can see neuronal loss. And the second animal model is a CAMTET mouse model. So between the two of them, you see pathologies of plaque buildup, and you also see neuronal loss. The whole focus here is putting cells into the hippocampus, which is ground zero for memory. And in these animal models, Dr. LaFerla has shown memory enhancement with the mouse cells and with the human neural stem cells. So to sum up, what we're doing is replicating the work that was done in a more controlled and more focused fashion, so that the quality of the work and the checks and balances would meet FDA standards for the filing IND.
Okay, very good. We're past 50 minutes here, so let me just ask one more question for Greg. I don't want to leave you out since you're on the call, but talk about the balance sheet quickly. You ended the year with about $30 million in cash. You do have two planned phase 2 studies you're going to move forward with in spinal cord and AMD. So with all of that, give us a sense of how far you expect your current cash will take you.
You bet. So, I think we entered typical with a lot of biotech pursuing, this type of innovative work. You have essentially a year of cash in your balance sheet. We had $30.6 million, I think, when we entered, and I think the guidance that we gave on our quarterly call was that we expected a net cash usage somewhere around $30 to $34 million at the absolute upper end this year. Slightly more than what we had last year, just due to the fact that we've got a lot more clinical activity underway, as well as a lot more activity associated with the IND for Alzheimer's.
Okay, very good. Well, to wrap it up, I'll just ask you, Marty, if there's anything else that you want to highlight about the company.
Well, yeah, I mean, I think you hit on it earlier on, Kay, when you said, data. I think data, there are two words that I would use in my summation. One is data, the other one is valuation. I think the current valuation is very attractive given the relatively near-term opportunities for significant data events. But to date, we've transplanted a total of 30 patients. We've only announced data on 13, 6 of which were in Batten's, 4 of which were in PMD, and only 3 of which were in spinal cord injury. So this year you're going to see a significant flow of data all the way through to release the final data early next year in the phase 1-2 studies. And then you're going to see more data come out in the phase 2 studies, which will start with interim analyses 6 months into the study when we've got about half of the patients in these control studies, and then the final data which will come out. So you're looking at a very significant transition where data will drive the story, data will drive the valuation, and we're very happy, willing, and able to let our data do the talking. Very good. Robert, I don't see any questions from you, but if you did have one, go ahead and ask.
If anybody had a question at the time, please press *6 on your phone and then ask a question. Okay, we have no questions at this time.
Okay, well, very good. Once again, I appreciate the management from StemCells being on the call with us today. Again, from our view, we think this is a really good time to do a little more work on the company if you're not familiar with it. To Marty's point, there's a lot more clinical data coming, and clinical data is what's going to drive the valuation of the stock. So I want to thank you guys for being with us and the callers with us today as well. If you need any more information about the company, please give me or Robert a call. And I believe management, you're always available to speak with investors if that's what they're interested in doing.
Absolutely. We would look forward to speaking with anybody that'd like to get further information on the company.
Okay, very good. Thank you, everybody.
Thank you. Thank you.
I want to thank everybody for joining us on our call today. If anybody needs a replay of the call in its entirety, please dial me at 646-362-6240. That'll complete our call today with StemCells. Thank you.